Hello, everyone. I thought I would read to you The Synthetic Nature of Biology. Um, this is from a chapter from August 2015. The Synthetic Life Preamble. The basic unit of life is the cell, with the capacities of genetic, hereditary, and evolution as unique hallmarks. For centuries, biology has been the science of life focused on the analysis of the microscopic. Uh, Membrane cells, tissues, etc., and macroscopic insects, animals, plants, etc., worlds. But it's gradually adopted synthesis as a means to understand biological systems since the late 19th century. In 1899, for instance, in a manner that is reminiscent of today's media, the Boston Herald newspaper reported sensationally the work of the U.S. German biologist uh, Jacques Loeb, and I'm probably going to butcher some of these names as we go through this, so you'll forgive me, I hope. As the creation of life, Loeb is known for his invention of artificial parthenogenesis. Embryonic development was induced by treating sea urchin eggs with inorganic salts. Loeb conceived of living organisms as chemical machines, and he aimed for a synthetic science of life, capable of forming new combinations from the elements of living nature, similar to the way in which an engineer sees his work as practical, useful, and controlled. If we also recall the synthetic method to understand life by French professor in medicine Stéphane Leduc, uh, the creation of a new species of by experimental evolution by the Dutch botanist Hugo de Vries, or synthetic new species by genetic engineering by the American botanist Albert Blakesley. We can see that ideas of synthetic life resounded as early as the 1930s. During the second half of the 20th century, the field of genetic engineering consolidated, allowing for the emergence of a new era in biotechnology. This was made possible by major breakthroughs, including one, the elucidation of the structure of the molecule responsible for heredity in living organisms, the double antiparallel deoxyribonucleic acid or DNA helix by James Watson and Francis Crick in the 1950s. Nobel Prize in Physiology or Medicine in 1962. Number two, the discovery of restriction enzymes by Warner Arbor, Daniel Nathans, and Hamilton Smith in the 1960s. They got a Nobel Prize in Physiology or Medicine in 1978. Number three, the application of the restriction enzymes for DNA recombinant technology by Paul Berg and colleagues in the 70s, a Nobel Prize in Chemistry in 1980. Number four, the development of technology for oligonucleotide synthesis in the 1980s, fundamental work for modern molecular biology, not yet awarded a Nobel Prize. And number five, the development of the polymerase chain reaction for the specific in vitro amplification of DNA by Carrie Mullis in the 1980s, a Nobel Prize in Chemistry in 1993. During these decades, two journal articles referred to the term synthetic biology, one to highlight the potential impact of recombinant DNA in biotechnological applications, uh, and the other to bring up relevance in the political debate. By 1980, synthetic biology was defined as the synthesis of artificial forms of life, but it was also considered synonymous to bioengineering by some scientists. During the 1990s, while designing synthetic molecules uh, became a common practice during biological research, especially in the US, the field of metabolic engineering was emerging. From a historical perspective, synthetic biology has been evolving from an old genetic one gene era towards a younger metabolic two or more genes phase, followed by current genome dozens of genes engineering era that will drive us to a biosystems engineering more than one organism future. In fact, with the increasing sequences of genomes from different species at the end of 1990s, the genetic program of complex living systems increasingly came to be regarded as digital, a view that had been common for some researchers working in the information technology sector much earlier. Since then, biology increasingly depends on computers and mathematics for analyzing huge amounts of DNA sequencing data. But it was not until the new millennium that the contemporary field of synthetic biology reemerged from a community of engineers interested in biology. Their migration into biology has enabled the application of engineering principles like design, modeling, abstraction, and modularity of circuits in living systems for useful purposes. The culmination of synthetic biology as an engineering discipline has been considered by some bioengineers to be in 2004 during the first international meeting of the BioBricks Foundation at the Massachusetts Institute of Technology, or MIT, in Cambridge, USA. Uh, Let me just see if I can enlarge this little. We have synthetic biology with bioengineering and biomedicine, gene and metabolic and genome engineering, synthetic genomics, bioinformatics, systems and quantitative biology, biocomputing, information theory, xenobiology, biometric chemistry, in vitro projects, protocells, origin of the research, biosafety and biosecurity, social science ethics, philosophy and religion, the do it yourself biology, and artists, designers, and architects. Interesting. All right, let's continue on. Sorry. 1.2 Contemporary Synthetic Biology. In 2014, a scientific committee on behalf of European Union suggested defining contemporary synthetic biology as the application of science, technology, and engineering to facilitate and accelerate the design, manufacture, and or modification of genetic materials in living organisms. In theory, synthetic biology is about engineering and not about science. But in practice, it's composed of different research tribes of scientists belonging to large interdisciplinary groups of biologists, chemists, engineers, computer scientists, physicists, mathematicians, scholars from the social sciences, humanities, artists, and do-it-yourself biologists. Figure 1 illustrates the main disciplinary synthetic biology tribes, but the reader is referred to a previous work where the first research network in synthetic biology were investigated. Importantly, an open dialogue among natural scientists, social scientists, and scholars from the humanities and other stakeholders has recently played an important role in shaping a more inclusive development of synthetic biology. Some practitioners of these fields use their own jargon and metaphors according to their agenda. For instance, while the assembly of parts into circuits, uh, parts being promoters into circuits, e.g., plasmids, and their implementation in a chasis bacteria suggests predictable engineering view of biology. It's evident that biology is not predictable, but rather context dependent. That is, what make a living cell are not the parts, but the interaction and relationships among them. The oracle at Delphi posed a question concerning a boat. If in time every plank has rotted and been replaced, is the boat the same boat? Yes, the owner will say. The vessel is not its planks, but the relationship between them. Another example is the metaphor of genome writing, referring to the full synthesis of a bacterial genome and its use for cellular reprogramming of a related bacterium. It is clear that we are able to copy DNA sequencing and print DNA synthesis genomes, but we are far away from writing and designing genomes de novo because most of them are not completely understood, or the function of many protein coding genes is still unknown. Among other limitations, absolutely, the diversity of the synthetic biology practitioners is vast, and each research tribe has an agenda. But instead of promoting particular agendas that could hamper the development of others, tribalism, it is important to critically assess the various approaches that will evolve into independent methodologies in the coming years of research. To this end, synthetic biology can be broadly divided into four main engineering approaches: top down, parallel, orthogonal, and bottom up. Top-down engineering. This approach aims to reduce the complexity of extant cells by comparing universal genes and deleting non-essential ones for constructing a minimal genome. These efforts rest on the idea about the existence of a universal minimal genome that gave rise to all living beings based on assumption of a unique origin of cellular life, so-called last universal common ancestor, or LUCA. Excuse me. However, recent research points to the possibility that the tree of life, composed of eukaryotic and prokaryotic cells, eubacteria and archaea, emerged from a community of primordial cells rather than from a single cell. Therefore, while the Holy Grail like quest for the minimal genome has become elusive, eliminating many non essential functions comprises the fitness of an organism and results in fragile genomes. Another approach in this area involves genome streamlining, whereby dispensable DNA elements, mistakenly dubbed junk DNA in the past, are deleted to stabilize genomes for optimal performance in many biotechnological applications involving microbes. These efforts coexist with the engineering of genomes and epigenetics in multiplex or combinatorial manner. Uh, Importantly, top down synthetic biology relies on comparative genomics as well as systems and quantitative biology. For an example of a top down engineering for the production of hydrogen as clean energy carrier, see the chapter by uh, I won't even, one shears in this book. The bottom up synthetic biology approach primarily aims to create protocells and find the transition between the non living and living matter by assembling three components. First, metabolism for extracting energy from the environment and to construct, salvage, and discard aged building blocks. Second, an informational program like nucleic acids to control the system. And third, a container bringing these components together for allowing their coordination. To achieve this, three important principles of self organization are taken into consideration: reproduction, replication, and assembly. Reproduction refers to the ability of a system to reproduce a similar copy of itself. Cells or hardwares reproduce, whereas replication happens when a system replicates an exact copy of itself. The genetic program, such as DNA or software, is copied. Assembly occurs upon aggregation of vesicles or containers uh, made of small droplets of organic molecules like lipids or liposomes, membrane like structures containing phospholipids. Protocell research coexists with other in vitro synthetic biology projects aiming at synthesizing minimal cells, metabolic pathways, or never born proteins, as well as imitating cellular processes such as cellular division and growth. Although no longer considered synthetic biology research by the current EU definition given above, this research deserves proper recognition as a synthetic biology research because it has potential impact on other synthetic biology areas such as metabolic engineering by the in vitro optimization of synthetic pathways. Parallel engineering or bioengineering. Uh, Parallel engineering research is based on the canonical genetic code that employs standard biomolecules, including nucleic acids and the 20 amino acids for engineering biological systems. It includes the standardization of DNA parts, engineering of switches, biosensors, genetic circuits, logic gates, and cellular communication operators for a wide range of applications in biocomputing, bioenergy, biofuels, bioremediation, optogenetics, and medicine. Most of those applications conventionally rely on the use of one or more vectors or plasmids for controlling the expression of two or more genes and or proteins. Plasmids are small, circular, double-strand DNA molecules that can replicate independently from chromosomal DNA, mostly found in prokaryotic, but also sometimes in eukaryotic cells. A large number of practitioners in this field are engineers aiming to abstract the complexity of biological systems into parts, devices, or systems whose interactions could be predicted according to the dictum, "What I cannot create, I do not understand," by Richard Feynman. The migration of engineers into biology resulted in the first model based design of genetic circuits, or circuit engineering, based on simple mathematical models such as the toggle switch and the repressilator. In some of those collaborations, biologists and engineers worked together with computer scientists to develop the next generation computer aided science or aided design, or CAD, software for engineered based synthetic biology. In summary, the main goal of bioengineers is to predict the behavior of living systems for the sake of safety applications, as Drew Indy says. Engineers hate complexity. I hate emergent properties. I like simplicity, and I don't want the plane I take tomorrow to have some emergent property while it's flying. Orthogonal or perpendicular engineering, also known as chemical synthetic biology, this approach primarily aims to modify or expand the genetic codes of living systems with unnatural DNA bases. And or amino acids. This subarea also relates to xenobiology, an emergent area at the interface of synthetic biology or exobiology, systems chemistry, and origin of life research. In the last decade, scientists have synthesized molecules structurally related to canonical bases of DNA to test whether those alien or xeno XNA molecules could be used as carriers of genetic information. Similarly, the DNA sugar deoxyribose has also been replaced by non-canonical moieties. The genetic code can also be modified or expanded to express information beyond the 20 canonical amino acids of proteins. One strategy uses orthogonal enzymes and a transfer RNA adapter from an unrelated organism to incorporate a given unnatural, non canonical, or xeno amino acid XAA into one or more proteins at one or more specific sites. The orthogonal enzymes are generated by directed evolution, a method that consists of repeated cycles of genetic mutagenesis, genotype, typic diversity generation, screening or selection of a particular phenotype trait, and amplification of improved variant for the next uh, iterate. I can't even pronounce that word. Iterative round. Dozens of XAA have been successfully incorporated into proteins in bacteria, yeast, and human cell lines. Dozens of XAAs, or xeno amino acids, have been successfully incorporated into protein in bacteria, yeast, and human cell lines, but also in more complex organisms like worms or flies. Directed evolution also enables the development of orthogonal ribosomes based on canonical DNA sequence changes to facilitate the incorporation of XAAs in proteins, or of mirror life biological systems endowed with biomolecules composed of enantiomers. Again, I'm stumbling over some of these words, of opposite chirality. Another method, dubbed experimental evolution, pushes microorganisms to incorporate XNAs into their genomes or XAAs into their proteomes by serial culturing. Orthogonal engineering based on experimental evolution also aims for engineering cells that can survive in asteroids, on the moon, and even on Mars. Although the changes at the DNA level are likely to be canonical, cells suitable for non terrestrial habitats could be considered as orthogonal life. For more details regarding the colonization of Mars with the aid of synthetic microbes, see chapter by C verso in this book. Synthetic life forms. Thanks to the technological advances in molecular biology, organic chemistry, and engineering in the last decades, the ease and speed of genetic modification has enabled the development of emergent genetically modified organisms. Various types of emergent GMOs. We have the genetically modified organism. We have genetic engineered machine. We have genetic designed organism, genomically edited organism, genomically recoded organism, chemically modified organism. For example, the engineering inspired designs of GMOs has resulted in the crowdsourcing of genetically engineered machines, while decreasing cost of synthetic DNA has enabled the reprogramming of genomically designed organisms. More recently, cutting edge molecular tools have allowed for the arrival of genomically edited organisms. Finally, evolutionary approaches have accelerated the development of not only genomically recoded organisms harboring expanded genetic codes, but also chemically modified organisms endowed with unnatural DNA bases or amino acids. In this work, the three first definitions are introduced, whereas the two latter ones have already been proposed by others. In the following sections, various types of GMOs, mostly microbial, are introduced together with methods for their development and potential applications in biotechnology. Since GMEs, GEOs, GROs are modified using synthetic DNA, all these fall under the GMO definition. CMOs can be considered GMOs or not GMOs, depending on whether their genetic changes are induced by genetic modification or serial cultivation, respectively. The main purpose of showing existing and emergent types of GMOs and non GMOs, i.e., or e.g., CMOs, is to illustrate the most recent efforts undertaken by particular research tribes to categorize them according to their engineering approaches. For an overview, see Figure 3. The biosafety and biosecurity issues that these synthetic life forms may represent are subsequently highlighted. In this manner, it's easier to explore the context of a particular GMO, CMO, regarding its history, origin, methodology, possible applications, and risk in order to enable a better understanding of the organism and possibly an accurate technological assessment. The definition of genetically modified microorganism is established, and there's a PDF for that. The synthetic nature of biology. So here's their little chart with all their modifications. The synthetic nature of biology. Genetically modified organisms are produced using old and modern genetic tools, genetic engineering tools based on standard nucleic and amino acids as building blocks via top down, bottom up, parallel, and orthogonal engineering. Engineering based GMO design and construction has resulted in the crowdsourcing of genetic engineered machines through the famous international GEMS competition below. In the bottom up approach, genomically designed viruses and organisms can be reprogrammed by assembling commercial synthetic DNA, which can be subsequently transplanted into living cells. The first GDO, or genetically designed organism, reported was Mycoplasma, whose genetic code, do not confound with genetic program, was not altered, in contrast to yeast, whose genetic code and program is being modified in order to accommodate unnatural amino acids. Genomically edited organisms are GMOs whose genetic material has been modified employing cutting edge molecular tools. Genomically recoded organisms are GEOs whose genetic code has been modified to accommodate unnatural amino acids and perhaps unnatural DNA bases in the future. Finally, chemically modified organisms are composed of unnatural nucleic acids or amino acids. Note that the overlap between top down and bottom up is not shown because no truly synthetic cell in which all components are synthesized in the lab and assembled into a living organism thus far exists. There is no overlap between engineering left and evolutionary right based on approaches because the first aims to predict function from structure, whereas the latter does not predict function because this is extremely challenging if non additive effects are taken into account. Genetically modified organism. The EU law defines genetically modified organisms as living entities whose genetic material has been changed in a way that does not occur in natural conditions through crossbreeding or natural recombination. Internationally, the Cartagena Protocol on Biosafety on, to the Convention on Biological Diversity, which includes 170 countries, legally considers GMOs as living modified organisms or LMOs. That is, any living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. The forerunners of genetic engineering, Paul Berg, Stanley Norman Cohen, and Herbert Boyer, developed molecular tools to introduce foreign genes into bacteria, providing the basis for the development of GMOs. In 1972, Berg combined DNA from the gram-negative model bacterium E. coli and the simian virus 40 in the exogenous plasmid or vector. The following year, Cohen demonstrated that DNA from the gram-positive bacterium Staphylococcus could be introduced and stably propagated into E. coli. This experiment gave rise to the first GMO. The next year, Cohen and Boyer reported the creation of the first E. coli endowed with a transgene originally from the South African clawed frog, uh, Xenopus, resulting in the first transgenic organism. By using plasmids as gene vectors, these proof of principle studies showed that genetic material could be transferred not only between closely related species but across unrelated ones. These experiments prompted Paul Berg to call for a moratorium on recombinant DNA technology to assess its risk, while Cohen and Boyer urged to file a patent for exploiting recombinant DNA technology. At the same time, many biotechnology companies were founded. In 1976, for instance, Boyer co founded Genentech, one of the first biotech companies that was able to engineer GMOs to produce human insulin and growth hormone, among other blockbuster substances, including alpha interferon or uh, erythropoietin. I'm not even sure how to pronounce that, and tissue plasminogen activator. Nowadays, many patients around the globe with diabetes 1, growth problems, and immunological disorders benefit from taking these hormones, sometimes on a daily basis. For his fundamental studies of the biochemistry of nucleic acids, with particular regard to recombinant DNA, Paul Berg was awarded the Nobel Prize in Chemistry in 1980, half of which was shared with Walter Gilbert and Frederick Sanger for their contributions concerning the determination of base sequences in nucleic acids. This brief introduction to the history of genetic engineering shows not only that scientists have been tinkering with the genomes of microorganisms for almost half a century, but also that this would not have been possible without the development of molecular biology, which was essentially the result of an interdisciplinary collaboration between biologists, physicists, mathematicians during the second half of the 19th century. During the 1990s, the field of metabolic engineering, two or more genes, emerged as an extension of genetic engineering, one gene, when the chemical engineer James Bailey realized that the microbial production of chemicals and antibiotics could be optimized if the metabolic resources of a cell could be adjusted, concluding that the emergence of a systematic paradigm for metabolic engineering will transform the present pharmaceutical, food, and chemical industries. The following year, Bailey analyzed useful molecules that could be successfully produced in microorganisms, while others proposed that the modifications of the carbon metabolic flux should occur only at the principal node of the primary metabolic networks to overproduce desired metabolites. These remarkable reviews clearly identified the metabolic engineering as an extension of genetic engineering, laying a foundation for the successful field that now allows the production of biofuels, pharmaceuticals, fine and bulk chemicals. J. Kiesling, most recent breakthrough has been the production of a precursor of artemisinin. Art. I'm going to just call it art because I don't know how to, I can't pronounce that correctly, which I believe that is wormwood. So they have, his most recent breakthrough has been the production of a precursor of artesian, an engineered yeast. It is chemically known as a sesquiterpene lactone and endoperoxide, the drug of choice to treat malaria, preferentially in combination with other derivatives. Like I said, that's wormwood. Every year, 500 million people become infected with this disease, and 1 million persons die of it in the developing world, mostly in sub-Saharan Africa, Southeast Asia, and Latin America. Every 30s, on average, a child every 30. So on average, a child dies from malaria worldwide. I'm not sure what, 30 seconds? Okay. Artemisinin is obtained from the leaves of the plant Artemisia annua. And please forgive me, people. I know I'm really butchering these words. Also known as sweet wormwood. Using its leaves, a simple tea used to be prepared to treat fever, including malaria and the traditional Chinese medicine, but it was not until the 1970s that it was rediscovered for modern medical science. Chinese scientists extracted an active compound for artemisinin known as, and again, I'm just going to, RTN, in the past, or King Haosu in Chinese. However, this drug remained largely unknown in the Western scientific community until 1979, when a review of its application was published in English language. In fact, it took a while for the rest of the world, and especially the World Health Organization, to discover the potential of the antimalarial drug. Unfortunately, it's been argued that there is a global supply shortage of artemisinin, owing to an increasing demand to the environmental factors affecting the harvest of a 14-week growing plant, primarily in Chinese and Vietnamese, but more recently in India and East African farms. Yet this is a matter of controversy. Artemisinin can also be chemically synthesized, but it's very expensive and non-affordable to most of the patients. To come up with another source of artemisinin, Kiesling and colleagues ingeniously inserted several genes from different species into the baker's yeast, Saccharomyces cerevisiae. I apologize. Which, fed solely by sugar and basic nutrients, is pushed to start a 3-day culture production of artemisic acid, a precursor that can be chemically oxidized by standard procedures to artemisinin. The process of producing artemisinin has been shortened to only 14 days, but this took Kiesling and colleagues many years of research with both successes and failures. For example, suboptimal expression of exogenous genes having different genetic code uses, or volatility of molecules were encountered, but these problems were solved by optimizing synthetic genes and reaction conditions. In the end, Kiesling's group was able to produce significant levels of artemisic acid by engineering the mevalonate metabolic pathway with a total of 11 enzymes from E. coli, the yeast Saccharomyces, and the plant A. annua in E. coli. The metabolic pathway engineered in E. coli was later transferred to a genetically stable yeast strain that can efficiently transport out of the cells up to 115 milligrams of artemisinic acid per liter of culture, following therefore a simple and inexpensive purification process. Recently, German scientists reported the development of an optimized system where the three-step chemical synthesis from artemisinic acid to artemisinin can be reduced to a more economical and efficient single step based solely on oxygen and light. This achievement should contribute to increasing the yields while reducing artemisinin cost. A dose cost of $2.40 United States dollars several years ago, but the alliance of the first nonprofit pharmaceutical company, Institute for One World Health, and the company co-founded by Kiesling's Amris Biotechnologies plan to decrease the dose cost tenfold to 25 cents, with an aid of $42 million from the Bill and Melinda Gates Foundation, and the cooperation of the French international company Sanofi Aventis for scaling up the industrial process. Thus far, more than 1.7 million of semi-synthetic artemisinin doses have been shipped to malaria endemic countries in Africa, including Burkina Faso, Burundi, Democratic Republic of the Congo, Liberia, Niger, and Nigeria. Sanofi, haven't we heard of them recently? And of course, Bill and Melinda Gates of Health Foundation is involved in it. And is this COVID really a cover up for malaria? Can't help but wonder that. Genetically engineered machines, or GEMs, are herein defined as GMOs that emerge yearly thanks to a crowdsourcing approach, the International Genetically Engineered Machines Competition. Undergraduate students pay a large registration fee for getting standardized DNA parts or biobricks. Biobricks can be thought of as type of DNA Lego. For discussion about the playing component in synthetic biology, see chapter by L. Litterist in this book. Most originated from the bacterium E. coli at the Registry of Standard Biological Parts in MIT to construct at home in less than a year biological systems composed of parts, devices, and systems. For example, the bacterium E. coli has been converted into eau de coli bacteria that smell like wintergreen or bananas, depending on the growth state. E. chroma bacteria that glow in different colors, or E. cryptor, a hard drive device to store information. The main differences between GEMs and GMOs is the former ones are built with biobricks and following engineered principles, whereas the latter ones are constructed without these two requirements. The idea behind this distinction is to emphasize the development of potential applications based on GEMs that can be predictable, in contrast to standard GMOs, where the endeavor is not per se attempted from the onset. Besides promoting creativity and innovation, iGEM serves as an international platform for fostering in young students self-confidence and awareness of the ethical, legal, and social implications of their synthetic biology project beyond the bench, i.e. human practices, or more recently, policies and practices. The first real iGEM competition took place in 2004 when 5 teams, Boston University, Caltech, MIT, Princeton University, and the University of Texas at Austin, participated. Participated. Depositing a total of 50 parts. The University of Texas team designed for the first biological photographic film with a bacterial lawn displaying the phrase, "Hello World." Since its first meeting, iGEM competition has grown steadily in the number of countries, teams, and delivered parts. Interestingly enough, most of the successful iGEM teams tend to avoid using the registry parts and prefer to deposit new parts, perhaps because a major portion of the parts have not been tested or do not work as expected. These facts illustrate not only the context dependency of biology and the importance of molecular relationships beyond synthetic DNA, but also the challenges that the iGEM competition will meet in the coming years. There's a need to better characterize the existing parts and to increase their quality, and there are additional issues regarding industrial applicability, team judgment, and research funding transparency. From all the winners at each competition, only a few projects have been published, and even less have a potential industrial application. This is why the application of engineering principles in biology based on standardized DNA parts is a challenging endeavor that requires understanding the complexity of gene networks and the variability of each cell with heterogeneous cellular populations. 2.3, genomically designed viruses and organisms. Genomically designed viruses and genomically designed organisms are viruses or living entities, respectively, that have been reprogrammed. Venters is a genomically designed organism. The genome of mycoides built in vitro by assembling purchased DNA fragments following by an in vivo assembly using yeast. The designed genome was then transplanted into the bacterium Capricolum, Mycobacteria capricolum, which upon replication in selective media acquired the phenotype of Mycobacteria mycoides. And again, I apologize, people. I'm really trying to pronounce these words correctly. The genome that JCVI SYN 1.0 contained 4 encrypted watermarks sequences indicated by numbers. I guess that would be their watermarks. Using a genome that was copied from nature into a computer and later on designed to be bottom-up synthesized by chemical means. In this work, these 2 new definitions are introduced to differentiate from other GMOs in which the genome is borrowed from existing organisms or viruses and is not entirely synthetic, top-down engineering. The first genome that was chemically synthesized was that of the poliovirus that infects humans, followed by that of the bacteriophage phi X174 that normally infects E. coli. The genome of the 1918 Spanish influenza pandemic virus was then synthesized, followed by other human retroviruses. All of these GDVs, genomically designed organisms, were capable of infecting cells, suggesting that chemically synthesized genomes of higher organisms would be functional if these could be inserted into living cells. In 2008, the Venter Lab reported the synthesis of the complete genome of 582,970 base pairs, or BPS, of the bacterium Mycoplasma genitalium, giving rise to the first GDO. In parallel trials, Venter's team transplanted the natural genome of M. mycoides and conferring the latter cell that the identity of the former upon cell division and genetic selection. Mycoplasmas are parasitic bacteria that cause respiratory and inflammatory diseases in humans. They lack a cell wall, but the reason why these bacteria were chosen as model organisms is because they bear the smallest genomes among all bacteria that can support cellular growth in the laboratory. After 2 aforementioned breakthroughs, the next logical step was to combine them. In 2010, Gibson reported the genome synthesis of M. mycoides genome and its transplantation into M. capricolum, reprogramming again the latter cell into the former, but the difference being that in the genome of the reprogrammed cells, there were 4 encrypted watermark sequences, indicating the name of 46 persons involved in the project, to an email address, a website, and 3 famous quotations. To live, to err, to fall, to triumph, to recreate life out of life. Robert Oppenheimer's quote, see things not as they are, but as they might be, and Richard Feynman's What I Cannot Build, I Cannot Understand. Venter himself called these the first synthetic cells whose parents are a computer. However, the computer did not create the genome sequence. It served to store the sequence retrieved from nature upon DNA sequencing. Nor are the cells synthetic, only their DNA, which forms about 1% of the cell dry weight, that is synthetic. Nevertheless, this experiment has been regarded as a defining moment in the history of biology and biotechnology because DNA controls the hereditary information, and this raises the possibility of controlling and understanding life by using synthetic DNA. The synthesis of genomes is possible via synthetic genomics as an established field that emerged with the technological synergies between synthetic organic chemistry and engineering for high throughput DNA synthesis. Synthetic genomes, in turn, has enabled the emergence of not only other genetically designed organisms, including the baker's yeast and the bacterium Vibrio cholera, but also tools and basic research for assembling efficiently synthetic DNA. Last but not least, synthetic genomes promises to revolutionize the medical sector by reducing the time needed for the production of synthetic flu vaccines as okay needed for the production of synthetic flu vaccines in case of pandemics from months to days. Another healthcare application may be the development of synthetic bacteriophages, bacteria killing viruses. Given the recent emergence of antibiotic resistance, a huge global public health concern. Phage therapy is an old treatment that dates back to more than 1 century, but with the discovery of antibiotics during the first half of the 20th century, its application remained limited in the Western world. Thus, there is a potential in synthetic genomics for developing innovative phage therapies, but limited host range and side effects of bacterial lysis remain, among other non-technical issues. Genomically edited organisms, or GEOs, are defined here as those GMOs whose genomes have been modified with advanced molecular engineering tools. Genome editing can be performed in small or large scale, as when respectively mutating a single nucleotide polymorphism to correct a disease genotype, like in human cells or sickle cell anemia, or genome regions in multiplex for the combinatorial optimization of metabolic pathways in microbes. GEOs are also GMOs, but the reason behind the differentiation is to indicate the methodological differences. GMOs are usually modified using natural or synthetic DNA sequences encoded in plasmids, but modifying the chromosomal DNA using or not plasmids in GEOs usually involves the employment of a synthetic DNA that is bought online from biotech companies. Wow. GMOs are usually modified using natural or synthetic DNA sequences encoded in plasmids, but modifying the chromosomal DNA in GEOs usually involves the employment of synthetic DNA that is bought online from biotech chromosomal modifications typically include DNA deletions or knockouts, additions, knock-in, or replacements that are crucial to fundamental research for understanding the function of a given gene, protein, and/or genetic element in a physiological context. The foundations of genome editing can be traced down to the discovery of the cellular systems in yeast and mammalian cells that repair double-stranded DNA breaks, or DSBs, that otherwise would be lethal due to cellular death or oncogenic mutations. The repair of DSBs is possible by the activation of homologous recombination, which is the copy and paste mechanism that requires an undamaged copy of the homologous DNA segments as template for copying the DNA sequence along the break. In the past, UV radiation, chemicals, restriction enzymes were used to induce double-stranded breaks, but these were random and could not be directed to predetermined sites. Nevertheless, the pioneering work of Mario Capici Martin Evans and Oliver Smithies led to a basic understanding of the HR mechanism for repairing DSBs. In 2007, they shared the Nobel Prize in Physiology or Medicine for their discoveries of principles for introducing specific gene modifications in mice by the use of an embryonic stem cell. Since the 1990s, new genetic tools have been developed for modifying genomes more precisely, including rare cutting, homing, nucleases, zinc finger nucleases, and transcription activator like effector nucleases, talins. But specifically, inducing DSBs, these tools enable high HR that can be used in basic biology and to correct various disease causing mutations associated with hemophilia, sickle cell disease, and other deficiencies. Although ZFNs or zinc finger nucleases and talons nucleases provide access to the most of the recent healthcare applications. Both depend on custom-made proteins for modifying each DNA target, which limits their use due to large costs when multiple genes are mutated and simultaneously, as is necessary in a more complex diseases involving multiple genes. Nonetheless, a new tool dubbed CRISPR-Cas9 has recently emerged for editing genes in multiplex with excellent HR efficiencies comparable to ZFNs and talons, but lower cost thanks to the programmability at the RNA level. Composed of clustered, regular, interspaced, short palindromic repeats, CRISPRs of DNA and CRISPR-associated genes along the genome, the CRISPR-Cas9 is an immune system that evolved in bacteria and archaea to combat hostile viruses and foreign plasmids by inducing site-specifically double-stranded breaks. Here, I'm going to scroll back up here. Let's look at this. The CRISPR-Cas9 technology has been used to modify the genomes of bacteria, yeast, fungi, nematodes, salamanders, frogs, fruit flies, zebrafish, mice, rats, plants, crops, pigs, animal and human cell lines, as well as embryonic stem cells. The Streptococcus pyogenes Cas9 nuclease in complex with a single guided RNA and its target DNA was made using the 3D crystal structure protein data bank file. So, wow. So they have been able to modify all of these things using CRISPR-Cas9. CRISPR-Cas is an immune system that evolved in bacteria and archaea to combat hostile viruses and foreign plasmids by inducing site-specific double-stranded breaks. And I just love this woman's name, Dudna, DNA. It's all the world's a stage, and sometimes it's so in your face. The type 2 CRISPR-Cas9 system has been used for deleting, adding, activating, and suppress target genes with great efficiency in many organisms. The technology has allowed the correction of genetic mutation of diseases such as cataracts, cystic fibrosis, as well as the development of cancer models in animal tissues, and more recently, the generation of human cells resistant to infection by HIV, the human immunodeficiency virus. In just 2 years, since the beginning of 2013 until the end of 2014, examples of the application of the CRISPR-Cas9 technology in both basic and applied research has been reported and reviewed in more than 1,000 publications. The revolutionary Cas9 technology has already been compared to the restriction enzymes and the PCR, essential tools in modern molecular biology research, because it promises to accelerate the editing of genomes across the medical, agricultural, environmental, pharmaceutical, chemical, and biotechnological sectors. Genomically recoded organisms, or GROs, were introduced by the Church Lab at Harvard, referring to microbes whose genetic codes have been recoded using advanced genomic tools like those used for the GEOs. Genetic code describes a set of rules relating to the order of three DNA bases, codon, and a responding amino acid to synthesize proteins across all life forms, thus establishing a universal link between information storage and execution. For example, a small peptide composed of the amino acids master can be coded at the RNA level by the codons AUG, GCC, AGC, ACC, GAA, AGA, but this order can be modified to synonymous codons AUG, GCA, UCU, ACG, GAG, and CGG with the same amino acid meaning master. In addition, non-synonymous ones can also be introduced when another codon, the amber stop codon UAG, is used to incorporate a non-standard amino acid X as follows: the master. In 1968, Hawley, Khorana, Nirenberg shared the Nobel Prize in Physiology for Medicine for their interpretation of the genetic code and its function in protein synthesis. In the same year, Crick called the genetic code a frozen accident, implying that no new amino acid could be introduced without disrupting too many proteins. Since the 1990s, however, the incorporation of XAAs into proteins has been thawing the universal genetic code. There are two basic ways to engineer the genetic code. The components involved in the synthesis of proteins are engineered by directed evolution to allow the recognition of specific XAAs. But in these cases, the genetic code changes are usually non-heritable, in contrast to the second approach, in which organisms, so far bacteria, are pushed to incorporate XAAs into their proteomes and via experimental evolution, resulting in progeny with heritable changes. Both approaches, nonetheless, could be combined to render offspring dependent on XAAs for survival. Genetically recoded organisms are relatively new organisms that were introduced by George Church and colleagues at Harvard by exploiting the mechanism of HR in microbes. His team built up a device that automates the process of gene delivery, targeting, replacement, as well as microbial recovery and growth, called multiplex automated genome engineering, or MAGE. Let's look at this. The universal genetic code in RNA format uses adenine, guanine, cytosine, and uracil as bases, whereas DNA uses AGC and thymine. The 20 canonical amino acids are encoded by 61 sense codons. Translation starts at AUG codon and terminates at stop codons UAA. UGA or UAG amino acids are arranged in according to physiochemical properties: polar green, nonpolar red, basic blue, and acidic pink. Methionine. I won't read all that, but you can read this for yourself when I put this link down here. But let's go on. Up to 50 genome alterations at nearly the same time, producing a combinatorial genomic diversity. In one instance, Church and Weiss researchers were able to make the bacteria E. coli synthesize 5 times the normal quantity of lycopene, an antioxidant, in a matter of days and just $1,000 in reagents. Although this machine could accelerate the field of metabolic and genome engineering for the microbe-based production of biofuels, pharmaceuticals, and other chemicals, it's very expensive and very challenging to know with accuracy what genes and or proteins to target for mutagenesis. Since 2011, nonetheless, Church and colleagues have reported 5 tour de force studies using MAGE with another application in mind. First, they exchanged 314 314 out of 321 UAG stop codons to synonymous the UAA stop ones in several E. coli strains, but with some technical hurdles. Second, after overcoming the technical difficulties, they were able to recombine all strains and delete completely the 321 UAG codons, as well as the protein that terminates protein synthesis at this signal for yielding the first genomically recoded organism, E. coli 321. This bacterium exhibited improved efficiencies for incorporating XAAs into various proteins and increased resistance against infection by the bacteriophage T7 compared to the parental E. coli strain MG1655. Third, they probed the limits of MAGE by eliminating the most rare codons present in 42 essential genes involved in E. coli translation as a means to emancipate codons for incorporating further XAAs. In this work, it was realized that all non-essential genes could be modified with synonymous codons without compromising cellular fitness. Finally, based on the previous work in the E. coli strain on one hand, Church and colleagues and others, and on the other hand, Isaacs engineered essential genes to be functional by the rational dependence of XAAs, thus imposing the cells to be metabolically dependent on the external supply of XAAs. Both studies were able to show that E. coli can be contained in physical isolation without undetectable growth in liquid media for up to 14 to 20 days, as long as the XAAs was not added to the culture. The authors argued that their strategy is a significant improvement over existing biocontainment approaches, and that it provides a foundation for safer GMOs that are isolated from natural ecosystems by reliance on synthetic metabolites. Beyond biocontainment, other applications of GROs would be the production of proteins endowed with XAAs for basic research and perhaps biocatalysis. Chemical modified organisms are living systems endowed with unnatural, non-canonical, or xeno building blocks. The CMO term was introduced by Marlier, whose work was highlighted by Acevedo, Roach, and Budiza, referring to E. chlori, where there are two basic approaches for creating CMOs. One strategy tackles proteins, the main executors of information, whereas the other one deals with the information carriers or nucleic acids. As indicated in the previous section, together with some genetic tricks, the cultivation of microbes in the presence of unnatural building blocks allows their introduction in lieu of canonical amino acids or DNA. In 1983, Bacillus subtilis strain QB928 was able to grow on 4-fluorotryptophan, a synthetic analog of tryptophan, one of the 20 canonical amino acids. 30 years later, the same microbe does not require TRP anymore for propagation, but only a 4-fluorotryptophan, thanks to its adaptation that was accompanied by a relatively small degree of genomic changes. This is the first CMO whose proteome has been chemically modified by experimental evolution. E. coli has also been shown to grow on similarly fluorinated TRP analogs, but it also has the potential for accommodating other XAAs into its proteome. CMOs with unnatural building blocks as genetic polymers have been also reported. DNA is composed of the bases adenine, guanine, cytosine, and thymine, each linked to a sugar, a deoxyribose, which in turn is connected to the two phosphate groups. But scientists have synthesized molecules structurally related to the components of A, T, C, and G to test whether these alien molecules could be used as carriers of genetic information, including iso-C, iso-G, K, X, Q, F, P, and Z, NAM, and 5-SICS. Likewise, the sugar deoxyribose has been replaced by threose, TNA, arabinose, ANA, and glycerol, GNA, hexatol, HNA, cyclohexene, CENA, fluoro, arabinose, FANA, etc. That's disturbing to me. Alternate Alternative genetic systems. On the top, the canonical DNA bases thymine, adenine, forming 2 hydrogen bonds, as well as cysteine and guanine, forming the 3 hydrogen bonds, are shown. In the middle and bottom layers, unnatural DNA bases that are achieved by classical synthetic chemistry are depicted using ZPVJKX, as well as ISO-C and ISO-G. See, that is disturbing to me. This is quite disturbing. Although most xenonucleic acids or XNAs are incompatible with living systems, very recently Hollinger and colleagues described the directed evolution-based engineering of polymerases, enzymes that copy nucleic acids, that were capable of synthesizing XNA from a DNA template and from DNA back to XNA. This work is very important because it showed that genetic information could be stored in diverse, unnatural polymers capable of heredity and evolution. Along the same vein, that using experimental evolution, Marler and colleagues practically replaced all T bases by 5-chlorosil in E. coli with concomitant dependence on this alien substance for survival. These modifications resulted in morphological alterations, yet the E. coli cells were able to survive, giving rise to the E. chlori, the first CMO bearing chlorinated genome and potentially endowed with a genetic firewall. More recently, Romsberg and colleagues reported the addition of the XNA pair NAM and 5-SICS to E. coli by introducing an algae membrane protein that allowed the transportation of the XNA pair into the cells, followed by its replication into a plasmid. Though this CMO has been regarded as the first living being bearing 6 bases as genetic code with many potential applications in basic and applied research, it should be mentioned that only 1 base pair per plasmid was present, corresponding to less than 0.0001% of the total genetic content of that bacterium, which is about 1% of the total cell dry weight. Thus, the in vivo replication and propagation of a truly 6-member genetic code controlling some biological process remains to be shown. Several applications of orthogonal engineering have been envisaged. Basic research on artificial genetic information has already resulted in a more accurate diagnosis of viruses in patients. The inclusion of the bases ISO-C and ISO-G into branch DNA assays, which detect pathogenic NAs, significantly improves the signal to noise detection ratio. These assays are important because the viral load is critical to determine the amount and type of drug necessary in patients with HIV and hepatitis C, 2 major health problems with 35 and 180 million cases worldwide, respectively. Using these assays, about 400,000 patients per year can be assigned to a more appropriate treatment and personalized medical care. Similar types of research or other XNAs are being pursued for diagnosing cystic fibrosis, severe acute respiratory syndrome. Similar types of research on other XNAs are being pursued for severe acute respiratory syndrome. Synthetic amino acids have also found applications in human health. Clinical trials using human growth hormone containing a modified XAA recently demonstrated that the required safety, efficacy, as well as increased therapeutic potency and reduced injection frequency in adults. In scientific research, while proteins endowed with XAAs have become invaluable tools for unraveling complex cellular processes within living cells, unnatural enzymes have potential applications in biocatalysis. That is the use of enzymes and or microbes to synthesize useful compounds like food additives, flavors, fragrances, fine chemicals, biofuels, bioplastics, biomaterials, and other molecules. In February 1975, scientists called, this is biosafety and biosecurity. In February 1975, scientists called for a worldwide moratorium on genetic engineering, given the concern of the potential accidental release of GMOs into the environment. Upon the moratorium, some institutions stopped any kind of genetic engineering research while others took the lead. deterring basic science and the development of innovative products in certain regions of the world. Nevertheless, the moratorium was critical because it allowed the planning of an international meeting in the Asilomar Conference Ground in the Pacific Grove, California, USA, where scientists agreed that genetic engineering should continue under stringent guidelines. This important meeting allowed setting down international guidelines on research involving recombinant DNA, giving rise to physical containment cautions for biological agents and safety levels 1, 2, 3, and 4. In the Asilomar meeting, scientists themselves took the initiative to come together, given the exposure of synthetic biology to the media and concerns about harmful intentional or accidental misuses of synthetic microorganisms. Several groups of bioethicists and members of non-governmental organizations have suggested another Asilomar meeting to discuss the responsibility of synthetic biology to research to society. Likewise, debates on synthetic biology under the Convention on Biological Diversity have considered a potential moratorium on the release of synthetic organisms, cells, and genomes into the environment. Although these debates were triggered under the publication of the first GDO by Venter and colleagues in 2010, biosafety concerns can be justified by the earlier synthesis of pathogenic viruses such as the deadly 1918 H1N1 Spanish flu, which killed an estimated 50 million people, that was resurrected from archaeological samples, or the polio virus, which was synthesized even without a template. Even though it's been argued that GDVs offer an opportunity to better understand infectious diseases, these pathogenic viruses also pose potential biosafety risks to the health of the laboratory workers and the public. Two incidents in 2014 at the Centers for Disease Control and Prevention in Atlanta, USA, where the Spanish flu virus was synthesized, illustrate this. Samples from a low virulence flu virus were contaminated with the lethal H5N1 avian flu strain. And bacteria causing deadly anthrax were not properly inactivated before their transportation to a lower biosafety level laboratory, potentially exposing staff to the dangerous pathogens. Beyond Asilomar, whereas biosafety deals with the inherent capability of organisms or viruses to cause disease, biosecurity is mainly concerned with their misuse as biological weapons for bioterrorism. Bioterrorism is nothing new. The intentional release of pathogenic toxins, viruses, bacteria, fungi, and insects is a mode of biological warfare against humans and animals that many countries have used in the past. For example, during World War I, a German secret agent traveled to the USA to infect horses with glanders, a severe infectious disease caused by the bacterium Burkholderia, which provokes respiratory ulcers, septicemia, and death in horses, donkeys, and mules. For this reason, the Geneva Protocol was introduced in 1925 to prohibit the use of chemical and biological weapons in an international armed conflict. Later on, the 1972 Biological Weapons Convention and the 1993 Chemical Weapons Convention were introduced to regulate the production, storage, and transportation of biological and chemical arms. Concerns over mishandling of harmful biological agents are nothing new to synthetic biology. The CDC in Atlanta, USA, besides harboring the synthesized Spanish flu virus, also possess dangerous human pathogens such as Francisella tularenus, and Yersinia pestis, Bacillus anthracis, and to have considered had to consider their potential misuse. Yeah, it goes beyond. We have to create these things. We have to pull up the 1918 Spanish flu and recreate it because somebody else might do that. So we have to study it and create these Frankenstein things because someone else might do it. I mean, their logic is just complete insanity. Anyways, I apologize. Indeed, the international regulations for gene synthesis were lacking for several years. It was only after 2007 when a journalist ordered online from a biotech company synthetic DNA of the smallpox virus to his home that the synthetic biology community proposed a set of policies related to the processes involving nucleic acid synthesis. Among the new proposed policies is a first set that applies to the firms supplying synthetic DNA. Using a database of pathogenic genomes and sequences of toxin genes, the firms should use special software to screen orders for potentially harmful DNA. A second set of proposed rules is aimed at regulating the purchase of DNA synthesizers and the reagent used in synthesis by enforcing the regulation of the machines or the distribution of licenses to purchase specific chemicals needed for DNA synthesis. The new synthetic life forms beyond existing regulations. Beyond contributing to the development of rules and regulations, the research community has also played a part in shaping biosafety and biosecurity policies with the usual conjecture that what is natural is potentially more dangerous and uncontrollable than what is artificial. Because organisms close to their progenitors are pre-adapted to their native environments, it's logical to assume that GMOs, GEMs, GEOs could pose a major threat uh, would pose a major threat. Uh, okay, that doesn't make sense to me. Uh, than GDOs, GROs, and CMOs if all these were accidentally released in the environment. However, since the latter organisms use or potentially can use synthetic building blocks as constituents that could only be synthesized in the lab, they would be quickly out competed by other organisms if the chemicals are correctly disposed. Therefore, orthogonal and bottom up engineering should also follow the guidelines that chemistry labs have established for chemical disposal. Rather, or together those for biosafety labs involving GMOs, which is the norm for top down and parallel engineering approaches. Given that the synthetic biology is an extension of genetic engineering, current legislation on GMOs also applies to GEMs, GDOs, GEOs, GROs, and those CMOs that have been previously modified to incorporate unnatural DNA bases and or amino acids at both the EU and international level. But there have been doubts about whether the current risk assessment procedures would be overburdened by the increasing pace of genetic modification, which is the case for GDOs, GEOs, and GROs. In consequence, the latest EU recommendations call for research to improve the ability to predict the behavior of complex engineered organisms and for the development of additional approaches, including genetic firewalls based on a non canonical genetic material. Genetic firewall data are missing. It has been advocated that unnatural DNA would prevent information exchange between natural and synthetic organisms by acting as a genetic firewall. Indeed, Church and colleagues showed that the first E. coli GRO was capable of resisting to some extent infections by the T7 bacteriophage because their genetic codes are not compatible. However, the GRO infection by the virus was only slightly attenuated compared to the parental strain, implying that there are other factors that account for the lack of complete immunity. So, how many changes are needed in the genetic code to render a GRO completely immune to viruses without affecting its fitness? Given the high mutability of viruses, would it be possible that these could hijack new genetic code if sufficient time is given that in a non-controlled environment? If GROs and CMOs are not able to survive outside the lab, what would be the response of a natural organism to their unnatural building blocks? On the other hand, if a GRO or CMO dependent on XAAs manages to survive the environment by any means, would it pose threats to other natural organisms? And if so, what kind of threats? Would GROs be more robust than CMOs in case of an accidental release in the environment? Are there other means of cellular communication beyond horizontal gene exchange that GROs or CMOs could use to persist or even proliferate? Clearly, many questions remain to be solved to show that a genetic firewall could be used as an efficient means to separate the natural and the synthetic world, especially when it is believed that XAAs-based biocontainment could be a means to eliminate the fears of the public when it turns to apply GMOs in agriculture, medicine, and environmental cleanup. genetically modified humans. In March 2015, two groups of scientists called for a moratorium and for a framework for open disclosure on any experiments that involve genome editing in human embryos or cells that would give rise to sperm or eggs. The articles were responses to some rumors by referees who reviewed the work by Chinese scientists describing the use of CRISPR-Cas9 system in human embryos, who fear it could trigger a public backlash that would block legitimate uses of the technology. The main problem of the technology are the secondary effects, off target and on target mutational events with unintended consequences that are not yet completely understood. For example, monkeys have been born from CRISPR edited embryos, but at least half of the 10 pregnancies in the monkey experiments ended in miscarriage. In the monkeys that were born, not all cells carried the desired changes, so attempts to eliminate a diseased gene might not work. Despite this, the rumors of the referees became truth after some months. Work concerning genomic addition of human embryos was recently published, but the authors argued that it was necessary to illustrate that the CRISPR-Cas9 technology is still far from clinical applications. During a meeting in Napa in early 2015, Berg, Church, and other scientists called the urgency for a framework to discuss openly the safe and ethical use of CRISPR-Cas9 technology to manipulate the human genome. The proposals for regulating germline engineering broadly include: 1, the discouragement of any attempts at genome modification for clinical application in humans in those countries where it's allowed. Some countries don't allow this kind of research or regulate it tightly. 2, the encouragement of transparent research to evaluate the efficacy and specificity of genome editing in human and non-human models relevant for gene therapy, as well as the implementation of standardized methods to determine frequency of off-target effects of physiology of cells and issues upon genome editing. Number 3, the creation of forums for the exchange between scientists, bioethicists, government, interest groups, and the general public to shape policy while discussing not only the risk and benefits, but the ethical, legal, and social implications for curing human genetic disease by genome editing. And I would just say that, right now with this rollout, Operation Warp Speed, and all these technologies that are using the RNA interference technology, which is literally going to modify us and make us genetically modified organisms, or genetically recoded organism. One of the many little whatever you want to call it, genetically modified, genetically recoded. And yet fact checkers are still saying that this stuff will not edit our DNA. It's just, it's beyond me how people can't see this for what it is. The fundamental issue with human germline engineering is that beyond treating genetic disorders such as Huntington's disease to eliminate human suffering, designer or genetically modified babies could be likewise engineered, facilitating the arrival of new eugenics era. Although human germline engineering is banned in several countries, not including the United States, a few labs and companies, primarily based in the United States, are working in this research line. One of the plans is to edit the DNA of a man's sperm or a woman's egg and use the cells in an in vitro fertilization clinic to produce the embryo, followed by its implantation into a woman's uterus to establish the pregnancy of the fetus. Another strategy that promises to be more efficient aims to edit stem cells, which divide rapidly in the lab and then turn them into a sperm or an egg. Although the CRISPR-Cas9 technology is still too immature to offer babies a la carte, 15% of the adults in a recent survey indicated that it would be appropriate to genetically modify a baby to be more intelligent. Besides engineering of a more healthy and intelligent baby, some transhumanists think the human genome is not perfect and that it could be engineered not only to protect against Alzheimer's disease, but to create super enhanced individuals to solve complex issues like climate change. Gene drive engineering. Will there be a meeting? The willingness of many scientists to engage in public discussion with other scholars shows that there is an awareness of the potential negative effects of emerging technology that has not been shown to be mature for clinical applications, especially when dealing with the delicate topics such as human embryonic stem cells. However, Less concerned scientists not working with the human stem cells have already devised a plan to create an autocatalytic genetic system based on CRISPR-Cas9 to spread mutated genes across populations of GEOs with high efficiency. For example, mosquitoes can be engineered to impair the transmission of genes involved in malaria and dengue fever. Uh, Beyond the potential benefits, the problem with this mutagenic chain reaction technology is that unintended off target mutations at essential or non essential genes could be triggered, thus spreading irreparable genetic defects or traits across natural populations of organisms and potentially driving populations with limited genetic diversity into extinction. The potential devastating effects of this technology in the wild have been recently warned by Lunzoff. Uh, Church commented, "This technology is a step too far." Yet he has filed a patent uh, for a more secure gene drive technology that he argues would offer substantial benefits to humanity and the environment by eradicating vectors that spread diseases, insect pests, and invasive species, without calling for thoughtful, inclusive, and well-informed public discussions to explore the responsible use of this current theoretical technology. In response to an ethical analysis on its regulation, it has been debated that the dual use potential of this technology raises concerns because gene drives carrying lethal toxins could be designed to eliminate particular human populations and attack their crops. In fact, Gurwitz concluded, just as exact technical instructions for making nuclear weapons remain classified for 70 years after the Manhattan Project, it is rightfully should, as they rightfully should, the gene drive methodological details do not belong in the scientific literature. No, these Frankensteins want to do this and then not even tell us what it is that they're doing. And their dual use gain of functions. Of course, they have to study it because some evil person might want to create this, or they might want to do this someday. So we have to do it before then. It's just that their logic it just drives me batty. However, Oy and Esveld disagree with that response, arguing that classifying information required to build gene drives cannot target potential misuses without also impeding development of defenses as well as environmental health, agriculture, and safety applications of CRISPR technology. These debates show the need of an international meeting involving all stakeholders involving all stakeholders. Yes, like all of humanity, we are all stakeholders in this, whether we own the money that they're making off the patents of this Frankenstein technology. These debates show the need for international meeting involving all stakeholders to regulate the development and deployment of GEOs endowed with gene drives that cannot be confined physically to a single country. In various surveys, synthetic biology has been perceived by society as an extension of genetic engineering, particularly in the production of GMOs. However, synthetic biology aims to avoid the same criticism that genetically modified plants and animals have triggered in various societies. Beyond the technology itself, in what follows, 7 topics are discussed in which the challenges, dilemmas, and paradoxes surrounding synthetic biology become obvious. Global social justice. Kiesling's artemisinin technology breakthrough is perhaps the most widely used example to show that synthetic biology, in this case the metabolic engineering tribe, can provide solutions to global health issues. However, social justice challenges have emerged because economies and employment in the South can be destabilized by synthetic biology carried out in the North. And in other words, although Kiesling's breakthrough has been welcomed by almost everyone in the synthetic biology community and other advocates fighting against malaria, there's a rising concern that the farmers who traditionally harvest and what could be losing their jobs, which would affect the families of more than 100,000 farmers worldwide. Furthermore, the introduction of semi-synthetic artemisinin could further destabilize the already variable prices of botanical artemisinin due to market fluctuations. This example illustrates the dilemmas and paradoxes that surround the development of synthetic biology in a globalized world. Now, we couldn't just grow more artemisinin and make sure that people have plenty of this natural herb. We have to make it synthetic. We have to come up with some bullshit reason why we can't grow it, that we have to create it in the lab with our synthetic. You know, um, these evil creatures, I think, are ruled by the bug, by the demon, by the AI, whatever you want to call it. It's all the same thing. Uh, the parasite running this place we call a planet. It has been argued that do-it-yourself biology will help in the worldwide democratization of synthetic biology in the same way that IT was democratized in the garages of computer hobbyists during the last century prior to the emergence of Silicon Valley and in as innovation hub. Right. Yeah. Yeah. If you have a brain cell left, then you ought to figure out that this wasn't come up with in the garage of some nerd. This literally came out of the government, the internet, and all of this stuff. Again, back to people being run by the bugs, which has an ultimate goal to attach us all and upload us all to the internet and have ultimate control. I digress. Do-it-yourself biologists or biohackers have equipped their garages, closets, and kitchens with inexpensive laboratory equipment. Although do-it-yourself biologists performed simple experiments in the past, such as insertion of a fluorescent protein into bacteria for producing glow-in-the-dark yogurt, it is expected that applications in health, energy, and environmental monitoring will emerge. In some instances, however, there has been a rising concern that homebrew drugs could also be produced by do-it-yourself biologists using synthetic microbes. This fear has been recently strengthened with metabolic engineers made yeast strains capable of synthesizing one of the various precursors of the opiate morphine, a precursor of heroin that has a high demand in the illegal drug market. Although this research intended to enable the long-term centralized and legal production of opiates for pain relief, engineered yeast for opiate biosynthesis could transform into illegal systems for criminal networks in USA and Europe, where drug demand is highest. Because yeast is extremely easy to be sent, a few dried cells per post would suffice to start a culture. Grown, water, and basic nutrients are only required, and process basic lab equipment such as centrifuges to extract any drugs using basic columns and resins for chromatography. Uh, Thus, the democratization, I'm sorry, of molecular biology might be more difficult to regulate than initially thought. Medicines, pharmaceuticals, fertilizers, pesticides, additives, cosmetics, plastics, cleaners, clothing, pigments, detergents, electronic parts, and many other essential products for human needs are conventionally produced by synthetic chemistry. In fact, chemical companies produce yearly billions of tons of chemicals from about 90,000 different substances. One goal of synthetic biology is to produce enzymes and microbes that can replace the production of many of those compounds because their precursors are usually obtained from fossil fuels and thus unsustainable. Hold on a second. I want to see if I can come up. I had come across an article. I can't find it. Where it was literally talking about yeast. It was talking about yeast being able to be programmed from the computer. I'll look for that and if I can find it, put the link down below. In fact, chemical companies produce yearly billions of tons of chemicals from about 90,000 different substances. One goal of synthetic biology is to produce enzymes and microbes that could replace the production of many of those compounds because their precursors are usually obtained from fossil fuels and thus unsustainable. A recent sampling of European rivers, however, illustrates the environmental damage that's been produced with toxic cocktails of hormones, pesticides, hazardous chemicals, let alone population declines of bees, birds, and other insects that are essential in the global food supply chain. In fact, irreversible damages of many toxic and non degradable and persistent chemicals on the immune, reproductive, endocrine, and nervous systems of many animals and likely humans have been found. To avoid similar environmental catastrophes using synthetic biology, proper legislation and regulation of the industrial production and waste of engineered microbes might have to be implemented from the onset. A challenge for synthetic biology will be to regulate and assess the long term effects of GMOs in the environment. To enhance biosafety biocontainment strategies, various genetic safeguards and mechanisms could be implemented in each engineered microbe together with a risk analysis and biosafety data sheet. The GMO debate. The GMO debate has existed since the Asilomar conference when biotechnology started to be regulated in general. But in the past 2 decades, it shifted towards risk posed by genetically modified food and crops owing to the food crisis in Europe during the late 1990s. Nowadays, GMOs are commonly used in industrial white and medical red biotechnology for the respective production of chemicals and pharmaceuticals and physically containments, but the genetic modification of plants and animals in agriculture, green biotechnology, has meant more resistance in society. Owing to the potential risk of GMOs for humans, animals, and the environment, there are different kinds of risks, real versus perceived, which, depending on the stakeholder, can vary significantly. Regarding the risk and semantics, uh, it's clear that the GMO debate is mostly associated to multicellular organisms such as plants and animals. Given the synthetic biology is increasingly targeting these multicellular organisms for genomic modifications, its acceptance by the public could be more difficult to gain. The main reason is that plants and animals directly affect human life, in contrast to microbes, which can affect human life more indirectly. As long as synthetic biology creates only new microbial life and does not directly affect human life, it will, in all likelihood, be considered acceptable. Beyond the potential of synthetic biology for solving global issues, society may only accept any kind of GMO, GEM, GEOs, GDOs, GROs, and CMOs if their products are labeled for the consumer in a transparent way, where the freedom of choice is given to consumers. One of the most common mistakes, media hype, metaphors, and promises, one of the most common mistakes is that scientists often make is to claim that they think that they're created something and that this creation they believe will be a panacea to humankind. First of all, scientists do not create because this word can be used in different contexts. Creation or creatio ex nihilo means to bring someone or something into existence out of nothing, and it's usually reserved for a divine force in religious terms that scholars in social sciences and humanities might use more often. In reality, scientists invent or design something that has not existed before by using something that already existed. They are closer to manipulado or manipulating and creating something out of existing parts than creating out of nothing. This is a subtle yet important difference. Scientists claiming that the creation of synthetic life have been accused of playing God in Frankenstein's footsteps. Yeah, with unfortunate consequences for the reputation of the field and its social acceptance. Perhaps the natural scientists do this on purpose to challenge the ethical and religious views of other scholars working on synthetic biology. On the other hand, overstating that GMOs, GEOs, EMs, GDOs, GROs, and CMOs will solve all human problems is not beneficial because exaggerating exactly into this direction generates mistrust in the public, as many emergent technologies have done in history. Hence, a lesson to synthetic biologists would be to be more cautious with their metaphors and promises if they want to gain public acceptance of their technologies. For a more insightful discussion on the impact of metaphors in synthetic biology, see the chapter by D. Faulkner in this book.
Semantics in the public. Synthetic biology is a term that's not well known to the public. In fact, recent survey in Germany confirmed this, but also revealed that people spontaneously perceive this field as an abstract, contradictory expression that they associated with interference against nature. How can nature be synthetic? Synthesis means to put together two parts to form a new one. This definition suggests that any minor modification of an extant organism would render it synthetic, which would be the case for all GMOs, CMEs, GEOs, GDOs, GROs, and possibly CMOs. However, a bacterium with a synthetic plasmid is not the same as a bacterium with a synthetic genome. So we should make a distinction. For example, a synthetic genome of a genetically a GDO represents about 1% of the whole dry cell mass. The cell reprogramming was performed using a genome borrowed from a related species, in which 0.1% genetic changes, watermarks were done. So why should this bacterium be called synthetic when the other 98.9% components are natural? In reality, this bacterium is a more natural than synthetic. Although genomes control gene expression, the truth is that genomes can be useful if there are proteins that can process their information, as it was the case for M. mycoids, JCVI SYN1. In contrasting example, when the genome of a cyanobacteria, Senectocystis, was cloned into the bacterium B. subtilis, genes from the former could not be expressed in the latter organism because of incompatibility or context dependency issues. Thus, synthetic genomes only represent minor parts of cells, in contrast to lipids and proteins, of which there are millions of molecules in a single E. coli bacterium, let alone other important components. A truly synthetic organism would be that which, in all the components shown, would be synthesized and assembled to generate a living organism. Indeed, synthesizing all the molecules of life was the dream of the famous German chemist Emil Fischer in the beginning of the 20th century, when synthetic chemistry experienced a major revolution almost a century after the breakthrough by Frederick Wohler in 1828. Urea synthesis, the first synthesis of an organic molecule in the lab. Thus, a real challenge for bottom up synthetic biologists will be to synthesize completely from scratch a living microbe. Given that top down, parallel, and perpendicular synthetic biology researchers mostly borrow microbes for tinkering, proving something by making small changes, the challenge will be to make the public aware that most of their research is not based entirely on synthetic organisms, but rather that synthetic DNA enables researchers to reprogram certain organisms for useful purposes. This endeavor should be facilitated clearly by distinguishing that DNA is not equal to life, but just another minor component of life, as important as the other ones. The resurrection of an extinct species has so far been reported for Celia, the last Bucardo, which passed away on January 6, 2000. It was resurrected for 7 minutes on July 30th, 2003 by nuclear transfer cloning. The germ cells had been frozen. Nuclear transfer involves the injection of genetic material to be cloned into an unfertilized DNA-free egg, resulting in cells that have the potential to divide when placed in the uterus of an adult female mammal. This is how Dolly the sheep was cloned from an adult somatic cell, but dozens of other species have been cloned despite the low efficiency of the technique. Despite nuclear cloning, it's been suggested that synthetic biology could cooperate with the biodiversity conservation community to protect or even resurrect extinct organisms. However, there are several technical and non-technical issues that first have to be resolved, and this is clearly reflected, even inventors being cautious. I have read too many articles that breezily discuss the reconstruction of a Neanderthal or a woolly mammoth with the help of cloning, even though the DNA sequences that have been obtained for each are highly fragmented, do not cover the entire genome, and as a result of being so degraded, are substantially less accurate than what is routinely obtained from fresh DNA. Further, if fresh intact DNA or software in the woolly mammoth or the Neanderthals were available, it would be necessary to have the appropriate cells or hardware that could interpret the genetic information. For example, when the components, a nucleus, cytoplasm, and cell membrane of amoebas of different species were combinatorially reassembled, the only visible or viable organisms were those whose components originated from the same strain. This old yet ingenious study shows that reprogramming of life goes beyond pure synthetic DNA. Although it's difficult to define life among scholars from different fields because no consensus has been reached from the up to 123 current definitions, it seems that life requires at least three components: a genetic program, a metabolism, and a container. For discussion about the concept of life, see the chapter J. Steisinger. Regardless of the definitions of any attempt to reprogram or resurrect life or construct lifelike systems from the bottom up, should consider establishing the connection among all these three components. Regarding the non-technical issues, it's useful to illustrate the pros and cons to better understand the dilemma of resurrecting extinct species with synthetic biology by quoting an expert. The de-extinction movement, a prominent group of scientists, futurists, and their allies, argues that we no longer have to accept the finality of extinction. The most persuasive argument is, quote, to appeal to our sense of justice. De-extinction is our opportunity to right past wrongs and to atone for our moral failings. De-extinctionists also argue that by resurrecting species, ecological functions could be recovered, increasing the diversity of ecosystems. However, the introduction of revived species could pose disease threats to native species in a manner reminiscent to the introduction of invasive species into new environments. In addition, some conservationists also worry that de-extinguished species would have a limited genetic diversity. In summary, one should ponder whether it's worthwhile investing huge amounts of human and financial resources in ambitious projects for resurrecting life, given not only technical and non-technical difficulties, uh, but also the paradoxes of our world. Devastating epidemics, hunting, habit loss, degradation caused by both human industrial activities and climate change are triggering unprecedented loss of biodiversity, with estimates of 500 to 36,000 species of amphibians, birds, mammals disappearing every year. Thus, instead of resurrecting life, synthetic biologists should help conservation biologists to develop innovative ideas for protecting the already thousands of endangered species. Conclusion: I have attempted to outline the most important areas of current research in synthetic biology in a general, inclusive framework, including different engineers' approaches. Almost all engineering efforts produce protocells, CMOs, and GMOs, some of which are developed by well-known genetic engineering methods, and others by using more sophisticated tools. For example, the production of genetic engineered machines is outsourced to undergraduate students every year across the world, with the advantage of developing creativity and innovation by younger generations for solving complex problems. Similarly, researchers working in synthetic genomics have produced genomically designed organisms as a result of basic research spinning off from revolutionary applications for the rapid assembly of synthetic DNA. More recently, the groundbreaking CRISPR-Cas9 tool, product of basic research, has allowed for the emergence of an impressive number of genomically edited organisms in less than 2 years to better understand disease models in cellular biology, as well as engineering multiple traits in microorganisms for the optimal production of drugs, biofuels, and chemicals. However, the use of CRISPR-Cas9 systems should be cautious, especially when engineering human embryonic cells as well as wild populations of organisms in the open environment. Finally, evolutionary approaches have accelerated the construction of microbial GROs and CMOs that promise to shed light on the meaning of and evolution of life by endowing living systems with unnatural building blocks. Importantly, it's any sort of genetic modification, such as single DNA base mutation, metabolic pathway optimization, or whole genome recoding of any of these synthetic life forms, will create a novel combination of genes and their products at the level pre and post transcription RNA and translation protein, thus triggering a new network of gene interactions at the transcriptome and proteome level in the host organism and across organisms, which are difficult to understand and predict using even the most advanced mathematical algorithms and technological tools. These phenomena, which fall under the scope of epistasis, emphasize the evolutionary complexity of gene interactions inherent to biology. For this reason, synthetic biologists should be aware of the limitations that must be overcome to predict the behavior of living systems. The purpose of illustrating the benefits, but also potential risk of these emergent life forms, is to provide scholars from the social scientists and humanities, as well as non-scientists, with a glimpse of what synthetic biologists are actually doing. The challenges, dilemmas, and paradoxes surrounding the field also showed that there are already big challenges in a globalized world. There cannot be solved exclusively by technological means. Whether synthetic biology will deliver health, food, or energy to societies with very different needs in the South and North, affecting an already polluted environment where biodiversity loss is an everyday phenomenon, then will be seen in the coming century. Okay, there you have it. I know that was quite a long read. Um, Hopefully you learned something with me about the synthetic nature of biology. Thanks, everyone.